Welcome to lecture number 12 on terpenes and terpenoids. In previous lecture, we have discussed the chemistry of phytol and abitic acid. Phytol and abitic acid belong to a class of diterpenoid. Similarly, vitamin A also belong to a class of diterpenoid. So in this lecture, we will discuss the chemistry of vitamin A. We will see the occurrence as well as uses of vitamin A. We will discuss the structure of vitamin A on the basis of chemical methods of analysis. We will see the applications of organometallic reagents as well as the Wittig reactions in the total synthesis of vitamin A. Vitamin A is also called as retinol because it is involved in the retina of an eye. Vitamin A is responsible for the vision of an eye, and therefore we will discuss the mechanism or the chemistry of vision of an eye. At the end of lecture, we will solve some problems related to the vitamin A as a revision exercise of this lecture. Now let us see from where vitamin A we get. Vitamin A is a nutritional molecule and there are various sources of vitamin A. Among that, the vegetables like carrot, broccoli, sweet potato, pumpkins are the major sources of vitamin A. Vitamin A is having multiple functions. It is useful for the maintenance of the human system as well as good vision. It is also important for the maintenance of the skin because it helps to maintain the health of mucous membrane and the skin. So many anti-aging creams contain vitamin A. Vitamin A deficiency leads to a disease called as night blindness. It also results in the dryness of the eyes as well as skin. So as far as our diet is concerned, vitamin A is a very important molecule. Now another source of vitamin A is carotenoids. There is a class of compound There is a class of terpenoid compounds called as carotenoids. Carotenoids contain 40 number of carbon atoms. It contains 8 isopreninates. And among these carotenoids, alpha carotene, beta carotene, gamma carotene, and the xanthophylls, they are considered as a provitamin A. So consumptions of this kind of the carotenoids in our diet, particularly from the carrot, these compounds are converted, that's the carotenoids are converted into vitamin A by an enzyme called as beta carotene 15,15-dioxinase. So another source of the vitamin A is the carotenoid molecules. Now let us discuss the structure of vitamin A on the basis of chemical methods of analysis. In chemical methods of analysis, first of all, the compound is isolated in pure form from the plants, and then it is subjected to estimations of carbon and hydrogen. From the estimations of carbon and hydrogen, chemists reached to the molecular formula of the vitamin A and it was found to be C20H30O. This molecular formula is a very useful information regarding the structure of vitamin A. Since it contains 20 number of carbon atom, we can assume that there are 4 isopreninates, and according to the isoprene rule, they may be joined through head-to-tail linkage. Now, since it contains 20 number of carbon atom, it belongs to a class of diterpenoid. Another information from this molecular formula we get is about the degree of unsaturations or the double bond equivalence. Double bond equivalence or a degree of unsaturation means how many double bonds or how many ring vitamin A contains. For a molecule CXH5OZ, the double bond equivalence is calculated by x plus 1 minus y by 2. So if you keep the values of carbon and hydrogen, the degree of unsaturation of vitamin A is found to be 6. So this 6 degree of unsaturation is a very important information regarding the vitamin A. We have to find out this 6 corresponds to how many double bonds, corresponds to how many rings present in vitamin A. Now after getting degree of unsaturation is equal to 6, let us find out the nature of oxygen. It contains only one oxygen, and for that this experiment was performed. Vitamin A was treated with para-nitrobenzoic acid. The reactions of acid, para-nitrobenzoic acid with the vitamin A results in the formations of ester. Now formations of ester by reactions of acid with alcohol is a characteristic reactions. So formation of ester is possible only when we assume the vitamin A as an alcohol. So alcohol on reactions with an acid will form an ester. This is a basic idea, and on the basis of that one can assume that the nature of this oxygen present in vitamin A may be the hydroxyl groups. So one oxygen present in vitamin A is of alcoholic type. Now let us find out the nature of unsaturation present in this vitamin A molecule. We have already proved that it contains 6 degree of unsaturations, and to prove the nature of 6 degree of unsaturations, we have to perform here a hydrogenation experiment. Vitamin A on hydrogenation in presence of a catalyst results in the formations of a molecule with formula C20H40O, and it is called as a perhydrovitamin A. So if you subtract the molecular formula of the hydrogenation product, that is C20H40O, from the vitamin A molecular formula, we see that it has consumed 5 molecules of hydrogen. So consumptions of 5 molecules of hydrogen by vitamin A, it means that it contains 5 number of double bonds. So out of 6 degree of unsaturation, vitamin A contains 5 double bonds. Now, if you do a chemical arithmetic, this is a very useful information to derive some other informations apart from the presence of double bond, and for that we have to do some chemical arithmetic. Now, the hydrogenation product formula is C20H40O, and its parent hydrocarbon will be C20H40, and this C20H40 corresponds to CnH2n. C20H40 corresponds to CnH2n and is a general formula for the monocyclic compound. Therefore, on the basis of this chemical arithmetic and the hydrogenation experiment, we can assume that vitamin A contains one ring. So, this hydrogenation experiment has given us two important points about the structure of vitamin A, that it contains the 5 double bonds and it contains the one ring. So, in this way, we have progressed a lot as far as the structure of vitamin A is concerned. There are 6 degrees of unsaturation, 5 double bonds, one ring, and the nature of oxygen is alcohol. Now with this information, we have to now find out more details about the skeletons or major structure features of the vitamin A, and for that degradation experiments were performed. There were two degradation experiments, one is a ozonolysis experiment and second one here is a cheminophore experiment. These two experiments are turning point of investigations as far as the structure of vitamin A is concerned. When the structure determination of vitamin A was going on, at that time chemists were having very good literature support of the chemistry of beta-enone. And beta-enone on ozonolysis gives the gyranic acid. It was a known idea to the chemist at that time. So vitamin A, when it is subjected to the ozonolysis, it forms gyranic acid. Similarly, beta-enone also gives the gyranic acid. So chemists thought that vitamin A must contain somewhere the beta-enone type of the skeleton structural unit present in vitamin A. So this is a very important information because this is the structures of the beta-enone, and beta-enone structure correspond to C13H20O. So if you subtract the molecular formula of beta-enone from vitamin A, the 7 number of carbon atoms are left. So out of 20 number of carbon atoms in vitamin A, we have accounted for 13 number of carbon atoms, nature of 13 number of carbon atom in vitamin A, and in the remaining discussions we have to account for the 7 number of carbon atoms. So ozonolysis experiment has proved that vitamin A contains one beta-enone in it. Beta-enone contains one ring, and we have already proved that vitamin A contains one ring. Now let us discuss another oxidation experiment. In this oxidation experiment, vitamin A is subjected to vigorous oxidations with the cheminophore, and there is a formation of acetic acid. Now formations of acetic acid is possible only when CH3 group is attached to the carbon-carbon double bond. We have to account for remaining 7 number of carbon atoms, and since here acetic acid is formed, there is some chances that we must have this CH3 attached to the carbon-carbon double bond. So isoprene rule, in combinations with this, we can assume somewhere in the somewhere in the vitamin A, we should have here carbon-carbon double bond, and to that CH3 group will be attached. Now, how many CH3 groups are attached and what are their positions with respect to each other? For that, one interesting experiment was performed, and it is nothing but the selenium dehydrogenation. Now, in this experiment, vitamin A is treated with acid, and vitamin A contains 5 number of double bonds, and since it contains 5 number of double bonds, under acidic conditions it will undergo some cyclization, some carbocation chemistry will be involved, and we will get some cyclic compound. And this cyclic compound, particularly here we will get here a fuse ring type of the compound. This compound on selenium dehydrogenation results in the formations of 1,6 dimethyl naphthalene. Now formations of 1,6 dimethyl naphthalene is a very important clue regarding the positions of the methyl group in the uh, vitamin A. We already proved that it contains a beta ion unit. We already proved that there are certain CH3 group attached to the carbon-carbon double bond. Now the question is that where they are present. So formations of 1,6-dimethyl naphthalene has proved that these CH3 groups are present with respect to each other at 1 and 6 position. So it's a 1, 2, 3, 4 and 5. So whatever the structures we are going to propose, these methyl groups are likely to be present at 1,6 position, 1,5 positions. So we have come a very long way as far as the structures of vitamin A, and now it's time to take the decision about the structures of the vitamin A. We have proved the presence of beta ion corresponding to 13 number of carbon atoms. We have proved the presence of 5 double bonds in vitamin A. This is corresponding to only 2 double bonds. So in the remaining cases, we should have a 3 double bond somewhere, and to that 3 double bonds somewhere, we should have the attachment of the methyl groups. And again, these methyl groups are at 1 and 5 positions. The nature of oxygen is an alcohol. So on the basis of all these chemical evidences, we reach to the structures of the vitamin A. Now we are in position to write it, and for that again the isoprene rule is again a very helpful. So on the basis of this chemical evidences, the structure of vitamin A is here as shown in this box. So this is a portion coming from beta ion and 1,5-methyl position here 1, 2, 3, 4 and 5, 1, 2, 3, 4 and 5. So here is again 1, 2, 3, 4 and 5. So 1,5 relationship between this methyl group is observed. So all these chemical evidences are in support of the structures of the vitamin A, and again one can understand the applications of isoprene rule here. The 4 isoprene units are joined through head to tail. Here is a head to tail, here is a head to tail. So on the basis of all these chemical evidences, we have proved the structures of vitamin A, which contains 5 number of double bonds, which contains the beta ion unit, and it's a alcoholic in nature. Now, once we reach to the structures of the vitamin A, it's time to convince ourselves by writing the equations involved in the previous chemical experiments, because at that time we were not much aware about the products. We only wrote the structural formula and then molecular formula. Now let us see here what happens during the hydrogenation. So five double bonds are completely converted to saturated hydrocarbon. Here is a monocyclic ring present. Then its oxidation gives us gyranic acid. Beta ion also gives us gyranic acid. Then its oxidation gives us a KMnO4. And this is the experiment where it requires some explanation. Under acidic conditions, some carbocation will be formed. This carbocation will be formed on this carbon double bond. It will form on this side, carbocation will form here, and then it will be attacked by the double bond. We will get a cyclation product, and this cyclation product undergo dehydrogenation with selenium. During dehydrogenation, this angular methyl group is lost, and one get here 1,5-dimethyl naphthalene. So on the basis of all these chemical evidences, we have proved the structures of the vitamin A. Now another proof for the structures of vitamin A comes from the synthesis. So synthesis of vitamin A is proposed on the basis of organometallic chemistry here, and it was reported in the year 1940. So vitamin A again can be prepared from beta ion. There are 20 number of carbon atoms, and out of 20, 13 are coming from here beta ion. So from beta ion one can reach to the structure of the vitamin A very easily, and for that we have to now offer 7 number of carbon atoms, and these 7 number of carbon atoms are coming from organometallic reagents. It is a very unique synthesis where there is a demonstrations of organo zinc compounds, organo lithium compounds, and the organo magnesium compounds for the synthesis of vitamin A. So here beta ion is treated with a reformist reagent. Reformist reagent offers 4 number of carbon atom. We get a 17 number of carbon atom as intermediate. Then it reacts with the methyl lithium. We get 18 number of carbon atom in this molecule. And later on, this Grignard reagent gives us a C20 number of carbon atom. And meantime, we will require some functional group interconversions in the form of oxidation, reductions, and hydrolysis. All these experiments, all these methodologies will be involved in the actual reaction sequence. Now let us discuss this synthesis of vitamin A on the basis of organometallic chemistry. So starting from beta ion, it reacts with the reformist reagent and we get a hydroxy ester here. It's a alpha beta unsaturated ester, and here is a tertiary hydroxyl group. Under acidic conditions, hydrolysis of this ester occurs as well as the dehydration of tertiary alcohol occurs, and we get here 4 conjugated double bonds here. Then it is treated with the methyl lithium. It is introduces one number of carbon atom here, and we are getting now here total 18 number of carbon atoms. So 4 is coming from reformed kit, 1 is coming from methyl lithium, and after this it is treated with alkynyl magnesium bromide, and we get a propargyl alcohol, tertiary propargyl alcohol. Tertiary propargyl alcohol on Lindlar's reduction H2PD BSO4, it gives us a vinyl ether, and it's a tertiary allylic alcohol. This tertiary allylic alcohol undergo acid hydrolysis, and enol ethers gives aldehyde, and it undergoes a dehydration here to form alpha beta unsaturated aldehyde. Alpha beta unsaturated aldehyde on reactions with the lithium aluminum hydride forms here vitamin A. So in this way, we have demonstrated here the applications of the organometallic reagents for the synthesis of the vitamin A. Here the key steps involved are organo-zinc compounds, methyl lithium, and the organomagnesium compounds. And the precursor for this is once again a beta-enol molecule. Now, after 1949, another synthesis was reported in the year 1958, after almost 10 years, and for that Wittig reaction was a key step involved here. This is a double bond which is constructed with the help of Wittig reaction. So if you do the retrosynthetic analysis of the vitamin A, we find that this is an aldehyde offering 5 number of carbon atoms, and this is a Wittig reagent. Wittig reagent can be prepared from allyl bromide. Allyl bromide will be prepared from allyl alcohol, and allyl alcohol is prepared from allylic reagent. It is a very common strategy in terpenite chemistry. Whenever there is allylic alcohol, chemists have employed allylic reagent as a key step to prepare the allylic alcohol. Allylic alcohol can be prepared from propargylic alcohol by reduction, and propargylic alcohol can be prepared from Grignard methodology, and this is again a beta-enone. Now beta-enone is easily accessible from an aldol condensations of citral plus acetone. So citral plus acetone gives a beta-enone, and from beta-enone we are reaching to the vitamin A. Here the Wittig reactions as well as the Grignard reactions are once again employed. It's not Grignard, it's a sodium in liquid ammonia. Now here beta-enone is treated with sodium in liquid ammonia in presence of acetylene. We get here tertiary propargyl alcohol. Tertiary propargyl alcohol on partial reductions gives tertiary allylic alcohol. Tertiary allylic alcohol undergo allylic reagent to form a allylic alcohol. Then on reactions with HBr it forms allylic bromide. Allylic bromide on treatment with the triphenyl phosphine forms a phosphine salt. And under the basic conditions, it reacts with an aldehyde to construct here a carbon-carbon double bond. To some extent, this is a stabilized halide, and this stabilized halide forms a transalkene. Plus, cis isomer is also formed, but is in a minor quantity. Now, trans as well as cis, they are separated with the help of chromatography, and then after separation, it is esterified, and later on, it is treated with the lithium aluminum hydride to form the vitamin A. So in this way, we have prepared the vitamin A once again with the help of a Wittig reactions as well as acetylene chemistry here by using sodium in liquid ammonia. So two syntheses of vitamin A are very interesting, and all the reactions involved are basic organic chemistry. Now the last portion is about the chemistry of vision. Vitamin A is responsible for vision process in our eye. Now, in retina of an eye, there are two types of the cell. One is a cone cell and another rod cells. Cone cells are responsible for color vision and for vision in bright light, whereas rod cells are responsible for vision in dim light. Now, this reaction sequence is taking place in the rod cells of an eye. In rod cells, vitamin A is oxidized to an aldehyde. It is called as a retinol, in presence of an enzyme called as retinol dehydrogenase. This retinol undergo isomerization in presence of retinol isomerase to form here a cis alkene. Isomerization occurs around 11 and 12 bonds, and it is called as a 11Z retinol. Now once this cis is formed, it is trapped by an enzyme called as opsin. This opsin is having an amino acids at 216 positions, and here is a side chain containing NH2. So this NH2 of the lysine forms sheep base with this jade retinol, and it forms a compound called as rhodospin. Rhodospin is called as a light absorbing substance. So when the light comes in contact with this rhodospin, what happens? It undergo isomerization. The cis is converted into trans. So conversions of cis into trans, this isomerization, when it occurs, an electric signal is sent to the brain where it is perceived as a virtual image. So this is main reactions responsible for vision of an eye. So cis is converted into trans, and here we get the perceptions of an image. And once this trans is formed, the geometry of the opsin molecule is disturbed and it becomes now a loose interactions. This geometry of this molecular geometry of the opsin is disturbed, and then this undergo a hydrolysis very easily. The process is called as a bleaching. One again get here retinol. Now this e-retinol, once it is formed, again the same sequence of reactions occurs. So conversions of here cis into trans is considered as a main reactions involved in the chemistry of vision of an eye. And here vitamin A is again considered, and that's why it's a very important molecule as far as our diet is concerned. So in this way, we have learned the chemistry of vitamin A. We have studied the structure of vitamin A on the basis of chemical methods of analysis. Uh, Beta-enone molecule on ozonolysis gives a zero-ionic acid, a very important clue. It contains 5 number of double bonds, then 1 positions of 1,5-dimethyl groups. We have proved the structure on the basis of this. Then the synthesis was proposed on the basis of organometallic reagents, and another was on the basis of Wittig reactions. So in this way, we have learned the chemistry of vitamin A. Now, after completing this discussion on vitamin A, now it's time to check our progress by solving some of the problems on vitamin A. So here some of the problems are listed. In this problem, a student is asked to write the structures of the vitamin A. Then applications of isoprene rule is to be demonstrated by identifying 5 number of carbon atoms of the isoprene unit and how they are connected head to tail. It belongs to which class of terpenite, and how it is proved that it contains 5 double bond and 1 ring. We have already discussed this in the initial stage of our lectures. Now second problem is related to the synthesis of vitamin A, where organometallic reagents are given here. These reagents are given in a random way, and we have to arrange them in an appropriate sequence to reach to the structure of vitamin A. And in this case, again we have to write down the structures of the intermediate compounds. And the last problem is related to the predict the product of reactions. Here Reformatsky reaction is involved. Here is a Lindlar's catalyst, partial reduction will take place, and under this is a Wittig reactions. So Reformatsky, Wittig, Lindlar catalyst, all of them are very important reactions from organic synthesis point of view. So students should undertake the practice to predict the product of such kind of the reactions. So in this way, we have completed the discussions on the vitamin A. In the next lecture, we are going to discuss about the beta carotene molecule. Thank you very much for watching the video. Thank you.